Did you do the activities? Yay! All right. Okay, so before I introduce the speaker today, I have a lost and found announcement. Does someone leave their beautiful prescription glasses from the October SUTS event? No? Okay. I'll leave them here at our closet, okay? All right, so welcome. Today we have Maddie Brightbill, who is a sixth-year PhD candidate from the plant biology program at UT Austin, and she'll be introducing everything about genetic responses of plants to warming temperature. So let's welcome Maddie.
Yeah, okay, right here. How is the volume? I know it was a little loud earlier. Is this good?
Yeah, it's good.
Okay, awesome. All right, hi friends, my name is Maddie. I'm a sixth-year PhD candidate at UT Austin. I study in the Sung Lab, which means that I'm both molecular bio and integrated biology. So this talk is going to incorporate some parts that are more like naturalist, like plant bio, and there's also going to be a good, decent bit of genetics in here. This talk is going to be a little bit more about something that's generally happening in biology right now than just my research, but we'll get into my research a little bit too. There's going to be a lot of the why and the what because the results are always changing. My goal for y'all tonight is to learn a little bit about what is currently happening in biology. There are currently a lot of people who are studying how plants are changing gene expression to respond to warm temperatures. It's not just me. And that's really exciting. So my goal for y'all tonight is not necessarily to just learn about what I do, but to learn enough about the field as a whole, so then whatever you hear about it in the news, you can say, oh yeah, was that Arabidopsis? Oh, you're talking about chromatin modification, for sure, for sure. So the goal is just for y'all to learn a few new things so you can seem smart in front of your friends. Sounds good? Sounds good. Awesome. Okay, so some quick takeaways. Very basics. Temperatures are rising. That's not just affecting us, it's also affecting plants. They need to respond in a way that's a little bit different. And today we're going to figure out how they can do it and how we can help. So, first I have a question. What do y'all do when it's hot out? Yes, front row. Yes, AC running inside, awesome. Anyone else? Maple, I see you. Make a bunch of lemonade, absolutely. I also heard someone say Barton Springs. I see a hand from David.
I sweat.
You sweat? Yeah. Yeah, anyone else? Yeah. So there are a lot of different solutions to this problem. I know for me, when I first moved to Texas from Virginia, it definitely took some adjusting for me too. But the punchline is, we can run two things, we can run from things, we can go grab water, we can find the AC. Plants can't move traditionally in the same way that we can. Also, I should get my, if I had my clicker, then I am not gonna have as much feedback, and will I? Okay. Sorry about that. So plants need to respond to a lot of different environmental stimuli just by staying in one place. So things like responding to drought, herbivory, so when things are trying to eat them, pests, mold, oxygen, CO2 levels, gravity, and for our intents and purposes today, temperature. So, a few quick things for why temperature and why flowering. Just anecdotally, I know for me, one of the first flowers that I really cared about was actually down here. Does anyone know what sort of flower this is?
Crocus.
This is a crocus, absolutely. And so we have these back in Virginia as well, and I know for me, something, I can't really remember what year it was, but at some point I realized that it was flowering earlier and earlier every year. And that was when I was in high school at least, maybe earlier. And bluebonnets, I'm not even gonna quiz y'all on that one because I'm sure y'all know. are flowering earlier and earlier every year as well. This past year, or in 2024, they flowered an entire month earlier than we expected them to. An entire month. So things are changing. Can anyone think of why plants might want to flower earlier in terms of warm temperatures? I saw two very quick hands in the front row. I'm going to go to you first because I don't want to be biased towards Maple. Yes, what's up? Absolutely, it might not be fit for flowering, or they might also see this environmental cue of, oh, okay, things are getting warmer, this period of cold has ended, so it's no longer time to be dormant, and the flower period is initiated. Also, it's incredibly useful to be flowering at the correct time and to be flowering quick enough if the situation is stressful, if the environment is stressful, so that way you can flower, get your seeds dispersed, and get out. So just wanting to make sure that that's all done. So I think I kind of covered this already. Sorry, the microphone, is this better or is this, nope, nope. I feel like there's a lot of feedback, sorry. Okay, so just very quickly, we're gonna be focusing primarily on the transition from bud development and flower initiation to flowering itself. And as you can see, temperature is an important step of that initiation. So for me, that is why temperature and why flowering and why it matters to me. Next, we're gonna get into something a little bit different. So previously I showed y'all some nice pictures of wildflowers, but that was a little bit of a trick too. Tonight we're gonna spend a lot of time talking about a plant called Arabidopsis thaliana. It is the plant geneticist's go-to plant. Technically, it is called the thale-cress. However, it is so unused as far as names go that when I typed in thale into this PowerPoint presentation, it came up as an incorrect spelling. So I'd like for us to all really quickly get used to saying the word Arabidopsis. So I'm going to say a sentence and I would like you to say it back to me. I planted some Arabidopsis yesterday. Absolutely. We use Arabidopsis to learn more about genes. Did you see that article about Arabidopsis in the newspaper? Awesome, thank y'all so much. So now we're a little bit more on the same page. I promise I'm not just trying to put you when I say Arabidopsis a lot. It's just that's just what we use. But technically, it is in the mustard family, that's Brassicaceae. Ooh. And its native range is technically Europe, Asia, and Africa. It's a very small part of Africa and then a slight bit of Europe and Asia as well. It has a very small native range. And I'll get into a little bit why that's the case later. But long story short, the people who started using it were Europeans, and so they kind of got to dictate what was going to become the model later. So and I'm glad we got some practice saying it. All right. So a little bit about Arabidopsis. This is just to get some anatomy down. The main part, Arabidopsis flowers once before it dies. It starts out with this rosette here, and then throughout the period of the floral transition, there is a few shoots that will sprout up and then grow. And then after the flower is formed, then you will have siliques. Siliques are where your seeds are, and then they will disperse. And then the flower itself is your normal reproductive organ flower. But yeah, so sepal, ovule, petals, just normal. Super easy to cross too, which is nice. So this is generally what it looks like. But once again, we're caring about the floral transition, so where we go from just the rosette to seeing this inflorescence for the first time. That's what matters to us. Also, just to look at different life stages again real quick. This is what Arabidopsis looks like throughout its entire life. Once again, we're mainly looking at the puberty here. So between this stage and this stage here, but they just go through one cycle of flowering and then they die. Okay, so we've learned a little bit about Arabidopsis. We learned how to say it. That's all fascinating. So why do we use it when we have beautiful plants, like, for example, orchids and other beautiful things we could use? Could anyone think of a good reason why we would want something that's a little bit hardier and like more stable and a little bit less expensive to use as a basic? I see a hand back there. Oh gosh, I'm so sorry about the feedback, y'all. Is it really bad? Okay, all right. Absolutely, you can subject it to a lot of different experimental conditions, and you don't really need to worry about it as much. It's very hardy, absolutely. And the other thing too that we've noticed is that Arabidopsis genes, which we'll get into later, we can actually apply those genes that we find, and it turns out there's a lot of similarities between plants like Arabidopsis and orchids. There is actually some research going on comparing the genomes of orchids and Arabidopsis right now. So we're learning new things, thanks to Arabidopsis. Arabidopsis is something called a model organism. So very quickly, we'll get into some other model organisms. This is a lot of text, so I'm going to give folks a second to read it. It does not all matter. But we see here some common model organisms are yeast, C. elegans, fruit flies, and mice. And you'll see that they have a lot of things in common. Can anyone very quickly tell me something that they see that's repeated a lot? It's all right, it's hard to see all the text. Yeah?
Ease of genetic manipulation?
Absolutely, ease of genetic manipulation is a major one. We'll also see things like simple growth, producing a lot of offspring very quickly. Small is often a big part of it. Quick reproductive cycles, these are all incredibly useful. So, Arabidopsis is like that too. It actually was the first plant genome to ever be fully sequenced. It only has 5 chromosomes that we need to worry about, so the DNA sequence itself is pretty small. It has a fast life cycle, so if you're mutating a lot of them, as we'll get into a little bit later, it makes that really easy, that turnaround time. And it's easy in general to genetically modify. In addition, there's a lot of cool things happening in Arabidopsis right now as a result. I know a few of you in the audience know about this experiment, but could anyone tell me what is happening here or what this experiment was? Hannah!
They got space stuff and they grew Arabidopsis in it.
Absolutely, they got soil from space or dirt from space. I don't know if it counts as soil, and they grew Arabidopsis in it.
Oh my god.
But yeah, and so they are running a lot of really cool experiments with Arabidopsis in zero gravity situations, and then also trying to determine if it can grow or if plants in general can be produced using dirt from the moon. And the answer is yes. It's difficult and not particularly productive, but it has happened. So there's lots of really cool things. That's just one example of cool things happening with Arabidopsis. So hopefully I've convinced you a little bit about this model organism, because we're going to move into something a little different. Now we're getting into genetics. Woo! The rest of this talk, or the main portion of this talk, the bones of it are going to be pretty genetics heavy. So I do have some terms that I want us to keep in mind before we keep going. So first, expression. I'll be referring to something called genetic expression, and I'll be defining it a little bit later on. But essentially, it's kind of like when you're expressing an emotion or an opinion or anything like that. It is not happening all the time. It is happening for bits and pieces, and I'll get into what genetic expression is specifically a little bit later, but that's just a little clue. Modification. There's 2 types of modification we'll be talking about tonight. The first part is going to be chromatin modification. So if you're familiar with chromosomes, I'll be talking a little bit about how that structure changes. Regulation, how gene expression is regulated, how it changes. Now we'll get into all of these more later. I don't expect you to have all these terms and definitions down now, but I just want to put them out there. Real quick, because we are actually going to get into genetic expression first thing. So, what I have here is something called the central dogma of biology. This is very simply, or to simplify it, that our DNA makes up the entirety of who we are. It is our entire recipe book, it is our entire cookbook, all of our information. But not all of our DNA is being used at the same time. In order to make the proteins that are the building blocks of our life, certain recipes are taken from our DNA and made into something called RNA that then become our proteins. And proteins just being like what makes up the organelles in your cells, and essentially when proteins are made, they make everything else, or almost everything else. So yeah, building block of life. I kind of alluded to this a little bit already, but I like to think about genetic expression and the central dogma like it is a cookbook. So, DNA is your cookbook. It has all of the things that are going to make you, or a plant throughout your entire life. But you're not going to use every single recipe all at once. You might not want to make breakfast for every single meal. You might not want a dessert at the middle of the night. I don't know, or maybe you do. We're riffing here. And RNA is the recipe you choose. And then protein is the meal that you make with it. So, we've talked a little bit about this transition between DNA and RNA, but why wouldn't we want to express every gene at once? Or why don't we express every gene at once? Can anyone think of why that might be? I'm seeing a hand in the front row, but I want to see if we've got anyone else, because we've participated a lot. But... all right, good to go, yeah. Absolutely, it takes a lot of time, it takes a lot of resources. Plants, for example, can't necessarily be putting all of their resources into both producing volatiles that will ward off different herbivores, while then also trying to produce fruit, while also trying to photosynthesize. There's a lot of different things happening all at once, and they might not be going on at the exact same time. If you think about it in terms of humans, you are not developing at the current rate, at the same rate now that you were when you were a baby or an infant. That slows down. You're not going to be expressing throughout your life all the same genes, and plants don't either. So, gene expression. But now we're going to get a little bit more into DNA, into that cookbook. When I say DNA, I typically think, and I feel like other folks might too, either about chromosomes, like you see here in this beautiful karyotype, or the A, G's, T's, and C's that make up our genetic sequence. I think that's what folks typically think about. But there is more that makes up DNA. It is not just these tightly wound chromosomes, and then when it's cell division is not occurring, then all of a sudden this loosey-goosey DNA. If your DNA was fully unwound, like it is here in this particular spot, it could reach from here to the moon. So it still maintains some structure. Got a lot happening on this slide, so we're going to go through it bit by bit. So, as previously mentioned, chromatin is that tightly wound structure that keeps all of our DNA wound intact. Chromatin is made up of nucleosomes that are wound around to create this tight spiral. Nucleosomes are comprised of two things: histones, which is a protein, and our DNA. And our DNA wraps around our histones in order to stay in place, stay all together. And what we're going to get into in a second is that, I kind of have these backwards. How tightly wound and how closely wound they are to each other, tight or loose, can change throughout can change. It's not going to stay the same the entire time. There are some bits that'll be loosey-goosey, like we imagine in the nucleosome or in the nucleolus, and there are some that will be a little bit more tightly wound, but it's never fully one, or it's not going to be fully just, there'll always be that structure. So, histones are used to change gene expression. But how do we choose what recipes are being made? Those recipes that if we're thinking about previously in that cookbook. We change those through something called transcription factors. This is a little bit of an aside, so you do not need to understand everything. Oh my gosh, oh no, I'm so bad at this, y'all, oh no. Okay, technology. So you do not need to be thinking about every single part of this complex. Essentially what is happening here is because we have these nucleosomes, these histones that are farther apart, then all of a sudden these transcription factors can bind and make this DNA into RNA, which can then become proteins, DNA, RNA proteins. So transcription factors choose what becomes RNA. It chooses our recipe. But chromatin modification, oh my gosh, every time, is going to change what actually can be read, what these transcription factors can actually bind to in the first place. And so I know this talk is going to be a little bit, it's a little bit heavy, maybe a little bit different than previous ones, so I am going to have us take a little stretch break, but it is a stretch break with a purpose. So if you feel comfortable, now would be a good time. Would love for y'all to stand up and we'll do a little stretching together. If you want to stay seated, totally fine as well. Okay, so I want to have us real quick illustrate what it would be like to be a piece of DNA that has a lot of space between its two histones versus a little bit of space. So first, we're going to be DNA that is open and ready for transcription. And I just want you to put your arms as far apart as possible. Just like this, just as far apart as possible. I'll do this so that way I don't get feedback. Oh. And as you can see there, if your body is the histone and your arms are the DNA, there's so much space to bind. If you feel comfortable even touching your neighbor's fingertips, look how much space there is. There's so much space. I know we're tight, but if my friends are getting rowdy, I think. But if they're all in this closed structure, if y'all could quickly do this for me instead, across the body, all of a sudden it's really hard to touch your next door neighbor's fingertips. There's a lot less space to bind. So that's just a quick way of wanting to demonstrate to y'all what this actually looks like. We can think about the stretching and the tightening. Y'all are more than welcome to take a seat. I hope you feel a little bit more awake. Okay, so we've talked about DNA, we've talked about RNA, we've talked about these recipes. Now we're getting a little bit back into plants, a little. I want to talk about my goal real quick. My goal has been, throughout my PhD, to find a gene that controls how plants flower under warm temperatures. I've been trying to find plants that, when bound to a specific location, can change how loose or how tight these histones are together, what this structure is like. And my goal has been to find a gene that helps control that and helps control flowering. So between two different temperatures, between 22 degrees Celsius, which is our regular normal temperature, and then 27 degrees Celsius, which is our warm temperature. 22 is going to be like your nice sunny and like 75 day. It's been a while since I've looked it up, like sunny and 75. And believe it or not, our 27 is only going to be about like mid 80s. Arabidopsis cannot deal with the heat the way us Texans can. So, oh gosh, I just have a slide that says why should we care with absolutely no notes on it, whoops. I know why I care about this is because I really care about the world that's coming to be. And we have done so many things to this world that have affected not only us humans and animals, but plants as well. And I think it's the least we can do to try to see if there's a way we can help them or at least learn more about them in order to progress. And how am I doing this? How am I trying to help them? We got to get a little bit into GMOs. Would anyone like to tell me what GMO stands for?
Genetically modified organisms.
Genetically modified organisms. I heard someone shout it out, but I'm sure the hands that were raised had a similar answer. And when we talk about genetically modifying an organism, just modifying a gene or a genome means just that. It's tweaking a piece of DNA. And we'll talk about the different ways that you can do it in a second. So I think a lot of times when we think about genetic modification, we think about crops. But it's not just crops, and I'm not talking about crops tonight. I am talking about experimental GMOs. This is a photo of me chowing down on some Arabidopsis, which you shouldn't do, and I only pretended to do. This one is heavily modified. So GMOs I'm talking about tonight, the genetics that I'm talking about tonight, is experimental modification and editing. So we're going to get into a few terms here, just like before, just to give y'all some keywords before we continue. First thing to keep in mind is modification and editing. So just things that we're doing to DNA to change it to learn more about it. Modifying it, editing it. Just like if you're writing a paper or trying to get something edited in your life. Absolutely. Interaction, how different genes relate to each other and how there can be different compounding effects of different genes. Genome, which we've touched on a little bit. Genome is just all of your genetic information that makes up who you are. That's going to be a word I start saying a little bit more, so we're just going to get that all sorted. And then the other thing I'm going to be saying a lot is wild type and mutant. Those are two different types of plants, your wild type and your mutant. Could someone please tell me what a wild type is? Or raise your hand. Yes. Yeah. Absolutely, it's the plant that occurs naturally. It's what we expect to see. Yes, and then your mutant kind of does what it says on the tin. It is something that has either been modified in a lab or just through nature to be different, to express different traits. We're already using our words from before. Expression, I think, was one of my previous ones. But so that's what we're getting into. A little bit more about GMOs, about how plants modify genes. The plant genome can be naturally edited in nature. Has anyone ever seen a tree that looks like this before? With this knot on the bottom. Sometimes when we go on a hike or something, you can see this tree that has a massive sort of tumor-looking thing on it. That is caused by a bacteria called Agrobacterium. And what that bacteria does is when it is on the plant, on the tree, on whatever, it will insert something called a TI plasmid, which is just tumor-inducing, pretty creative, into the plant DNA. It naturally does this. And then, much like a tumor, it will just create more and more copies of itself and grow and grow and grow. So this is a natural resource we have, Agrobacterium. So what we can do as plant geneticists is take Agrobacterium and just tweak it a little bit for our intents and purposes. Because we might not want that tumor-inducing, we might not want to just see giant tumors on trees, but we might want to change the DNA around a little bit. And so this is one way we can do it. I'm going to walk through step by step very quickly what the process of this is. Very quickly. This is a lot at once. This is the overall graph, but we're going to go through one at a time. Okay, so first we have our Agrobacterium like before. And what we do is we remove our TI plasmid. We just remove that TI plasmid. And then, as soon as we have that TI plasmid, what we do is we use something called a restriction enzyme to cut it open. So what we have now is just a TI plasmid from our agrobacterium that has been separated, and it is ready for something to happen. And what is that something? We can insert our own DNA fragment. We can take DNA that we have designed and use the same restriction enzyme in order to insert it into our plasmid. So now we have a plasmid with an inserted gene. Ta-da! That's the first half. On to the second. Now we have this agrobacterium with our new gene in it. And what it's going to do, oh goodness, every time, is it is going to be incorporated into the plant cell, just like it would normally in normal agrobacterium. And the way that you can do this, it's not anything super fancy. You can just straight up spread the bacteria onto the plant. What we do in our lab is we put it in some liquid, the agrobacterium in some liquid, then we take the plant that's flowering and we dunk it. It's super fancy and formal. But no, it's very easy for agrobacterium to do its job. And then finally, as soon as we have the agrobacterium exposed to the plant DNA, we can then grow plant cells and culture them, and then they will become a plant that is expressing our inserted gene or is mutated in the way we want it to be mutated. So once again, our overall diagram. But how would I use this for my project? If I am trying to find a gene that controls for this process, one of the things that I can do is I can try knocking out different genes, getting rid of them, and seeing what happens. So what I'm doing is I'm knocking out genes, or I did knock out genes, in order to determine their function. Because if something is not working, if there is a gene that no longer works, then we know what it normally does because it's no longer doing that thing. So that's what I did. Specifically, I had 10 different genes that I grew mutated lines of and then studied them for flowering time under 4 different conditions. And we'll talk about a few of them today. And luckily, I did find a plant that looked rather odd. So I'm going to explain what's happening here real quick. I have this mutant on the right that is called DOF 4.7. That just means that the gene DOF 4.7 has been knocked out. It is here in the first picture, and then here in the second one. And I have next to it our wild type on the left each time. Now what you can see is in our normal temperatures, our control 22 temperatures, our wild type is taking its time to flower, as it has every right to, but this mutant is flowering incredibly quickly, worryingly quickly. As soon as DOF is knocked out, there is no sort of control for holding this flowering back. And the really interesting thing is normally genes, if they're knocked out, they'll just do one thing consistently. Like they'll flower late both times or flower early both times. So what I was expecting to see was this 27 degree, this warm temperature also flower quicker, much quicker. But it did something even more interesting. It took forever to flower. It took forever to flower. It's doing the opposite thing in 2 different conditions. So what that means is, or what my hypothesis has been, is that this gene actively controls flowering time, not just in one of these conditions, but in both. It has a role in both. I'm going to get a little bit into what I've been doing since then, because this was preliminary results. But I'm sticking to flowering because we've done a lot of genetics, and let's do some phenotyping. Let's see what's been happening. So the first thing that I had to do was confirm that it was my mutation that actually was affecting flowering time. So what that means is it was possible that what we had mutated was random, and I hadn't targeted the gene that I thought I had targeted. So what I had to do in order to make sure I targeted the right thing was target the same gene 2 more times and make sure that it had the same outcome. So you don't need to pay attention to these names here. They're just names of different mutant lines. But essentially, for the first DOF mutant, the second DOF mutant, and the third DOF mutant, they are all having the same early flowering time in our 22 compared to our regular plant. And you can see that reflected in our data as well. Very quickly, the reason why we use leaf number here instead of just counting days till flowering time is because we care about that floral transition, that rosette leaf number. It's going to be very fickle if we just count number of days. And then another thing that I also found was that there's a similar trend between cold temperatures and regular temperatures. This is not something I expected, but it seems that consistently there is some sort of switch that's happening between our regular and our warm temperatures. So cold is the same as our control temperature for this mutant. These are early pictures because it takes forever to flower for 12 degrees in general. So you can't really see the flowers from here, but they're there. They're there. So these is the basics. I only wanted to leave you with a little bit of this during my talk because I've already been talking a whole lot about genetics. I didn't want to talk too much about my own research, but I am going to get into what's next, both for me and hopefully for plant biologists who are doing something similar. For me, things that I'm working on now, what does the DOF protein look like and what does it do? We know about the DNA, we know about the RNA, so we know about the cookbook and the recipe, but what does the actual product look like? What does it actually do? What is it interacting with? If it is regulating other genes, other things that work towards flowering, where is it binding and what is it interacting with? And if it is affecting histones, nucleosomes, the chromatin structure we've previously seen, how is it doing it? So those are questions that I'm working on now, what I'm doing now. And for other people in the scientific community, they are looking at other genes that might control flowering during warm temperatures in Arabidopsis. I feel very lucky to be a researcher at a time when biologists, plant biologists specifically, care a lot about what's happening with warm temperatures, and it's been a major topic, which is really cool. I'm glad that folks are working on that. And the other thing that's been happening is folks are trying to see if they can transform these results found in Arabidopsis, our model organism, to other species. So like orchids, like I was talking about previously. I've also seen this done in types of trees. Pretty much if you just Google Scholar and look up your favorite plant species, there's a good likelihood that there's some sort of connection being made. So that's what's happening generally. I would like to end us on a note that's a little bit dire, but I think it's really important. This is our temperature percentiles from this past August, as determined by NOAA. NOAA has determined the monthly temperature percentiles and temperatures since 1850. And what they do is they compare the current month's temperature against the same month's temperatures since 1850. Now, every year, every month of every year, I don't need to worry about updating this slide because I know that it's always going to be the worst month that it's been for that month, every year. And I've been doing the same slide since 2021, I think. Yes. I can't hear you that well from here because the microphone's sounding a little bit. Is it all right if we chat after? Oh, why is Antarctica so warm? Now that's a great question. Now that's something we should definitely get into later because that's not necessarily my entire area of expertise. But one of the things that people are really worried about is that places of the Earth that are supposed to be cold are getting warmer. And what all of this red means right here is not just that it's particularly warm, but it is comparing the level of warmth compared to other years. Yeah, exactly. It's much warmer than it has been. It's getting warmer every year, which is unfortunate, but it just means that as plant biologists and as biologists in general, as folks who care about our Earth, it's becoming really important to care about things like global warming and climate change. And I have a feeling that I don't need to convince anyone in this audience of that, but I think it is a good reminder. So that's what I'm working towards. I do want to say some quick thank yous to people in my life and to my lab. My lab is very small, so I have one picture with them. I am in the Sung lab. This is Dr. Sung. These are my lab mates. They're lovely. I have so much to thank for them. But I didn't want just one picture. I wanted to include some of my friends and my family too. So I just wanted to say thank you to all of my friends who came out tonight. And people who couldn't make it out. And I also wanted to say a major thank you to my family for supporting me along the way. Even though I'm in Austin now, which is kind of farther away, and I know they're watching too, and just thank you for that too. And I also, I didn't have a good photo of her with them, but I wanted to say a special thank you to my mom, because she is the best botanist I know. She is fantastic, and she is in this very exclusive gardening club. And this arrangement she did that. She has these fantastic arrangements, and she's the best botanist I know. And I just wanted to say a special thank you to her. Thank you all so much for your time. Have a good night. Oh, here, I'll leave this up for light. Just leave that up for light. All right, thank you all so much. Oh yeah, questions. That's a thing. Yeah. Do folks have questions?
Did you have a guess at which gene it might be when you were trying to map them out?
Yes. Long story short, we ran something called a ChIP-seq to look, and an RNA-seq to how to explain this well. So we were looking at genes that were specifically regulated by this other protein that we knew had an effect on warm temperature and flowering time. And so we were looking at genes that had different expression between regular temperatures and warm temperatures, and also between the presence and absence of that protein. So that was our clue. But even then, it was kind of a shot in the dark. And I honestly feel very lucky that I even found one that did work with flowering time, because there was no guarantee. With something like a genetic screening, there's not really a guarantee it's going to work out. And it was a lot of planting, a lot. Felt like a farmer for a little bit, which is not bad. But yeah, is there any questions? One in the very back? Yeah? Yeah. I cannot hear you. What's a thale? That is a great question. I wish I could tell you. I wish I was more of a naturalist. I do not know what a thale cress is, or a thale is. I know what a thale cress is. I'm so sorry. I promise I've done my fair research on Arabidopsis as it relates to the natural world, especially for this talk. I really tried to look into it as much as possible. Even scrounging up the pictures I did ended up being surprisingly difficult for Arabidopsis, because it's really just not something used in nature that often, because it has a smaller range and not that many implications. Yeah. Yes.
Is it an alpine plant? Is it like up and higher in elevation?
It is. It's slightly higher. It's like mountainous regions typically. Its range has expanded since people have started studying it. You can now find Arabidopsis in the US, which is a little concerning because it would be invasive and brought over by plant biologists. But yeah, it is like in mountainous regions, and yeah, absolutely. Yes.
I'm going to try to use this, and let's hope for the best. Can we give it a shot?
Yeah. How does it, so I want to make sure that I hear you fully. How does DNA turn into RNA? Turn into proteins? This is going to take a very long time, and I'm very excited about it. Long story short, there are proteins that already exist in a cell that can bind to, here, let me go back. And the proteins first will bind to the DNA, like they do here. But then as they bind, they will move along the strand of DNA. And as the strand of DNA, the original strand, separates from a double strand into two single strands, then two new pieces of DNA will be produced. I would love to draw this out for you, but it's a little bit, and drawing will be more helpful, but at the end, I would love to draw this for you. Yeah, any other questions? Yes?
You hold on one sec, and can you into the mic?
Hey, Abby, thank you so much for introducing us to the genes that controls the flowering process. And I wonder, so you introduced the idea that this is very important given our climate situation, and I wonder if you can go a little bit deeper into the implications, assuming that if we can understand a little bit more about the genes controlling the flowering circles, speckles, and how will this impact our lives?
Okay, so that is a great question. I'm very much on the basic biology side right now. So just trying to learn as much as we can as possible using model organisms, and then hopefully moving on. But there are a lot of biologists who are currently trying to figure out if there are homologs or homologous genes, similar genes in other species. There's some really cool research that's happening in trees right now, for example, to try to figure out if they have similar genes that are present. And I am not going to argue about the ethics of trying to modify, there are things that are a lot farther down the road, and I would say that it's not quite determined what will happen in terms of trying to modify things, or if that's even something that would be happening. But right now, on the basic biology side, we're just trying to find out all the information we possibly can about these genes that will help regulate. Fantastic question. Yeah. Anyone else? Well, thank y'all so much.
Thanks, Maddie. So thanks to everybody for coming. Next month on November 7th, we will have Luke Lauder. We'll be presenting on the nocturnal callers in Austin. So you can identify calls from crickets and frogs after his talk. Okay, awesome. Thanks everybody. Have a wonderful night and then have a wonderful weekend.